the pyruvate group, you have an aminotransferase again. You have a transamination reaction. You have glutamate pyruvate, and you get from glutamate you get alpha ketoglutarate, and the pyruvate it gets converted to alanine. So that's a very straightforward way to generate alanine. That's how alanine comes from pyruvate. Okay, everybody can make it. This is not an essential amino acid in human cells. So the carbon is the same. You start from 3 carbons of pyruvate, you get 3 carbons of alanine. If that was the only way, however, to make alanine, things then we should have been able to figure it out, because you can go and knock out that transamination reaction, that enzyme, and then you shouldn't be able to grow without alanine in your diet. And that has been done in microbes. And to this day, no one has been able to make this mutant an auxotroph, meaning that you have to give it alanine in the diet, which means that there must be other sources of alanine in the cell. Again, this reaction is shown in most textbooks as how we get, we and every other cell gets alanine made, but it turns out it's not essential for life. That's what it says, and it gets back to the genetics that, when in doubt, knock it out. When people go and knock it out, it turns out that it's not essential. So there must be other ways that we get alanine. So that's not the only one. For the rest of the pyruvate family, so alanine was straightforward. What's the rest of the pyruvate family? Sorry, valine, isoleucine, and leucine. So those differ in carbons. Okay, these are related amino acids. They differ in carbons, and you get what you put in. These are 6 carbon units. Isoleucine, valine has 5 carbons. So how does that happen? Well, you start, remember the principle again that when you use pyruvate to add, let's say 3, you may lose 1 because you lose a carboxyl group, and that's important to drive the equation forward. So that's really how it happens. You start with oxobutanoate in this case, you have pyruvate in this case, and you keep going down these reactions, okay, shown schematically here, to get you to the 6 carbon unit. So you combine a 4 carbon and a 3 carbon unit before you can get to the 6 carbon unit. 4 plus 3 is 7, you lose the 1 carbon and you got the 6 that you want. So, and that's all I had to say about this. You remember this thing that, again, you have to decarboxylate, you have to lose a carbon at some point to drive carbon-carbon formation. This is very similar. Going to valine. Okay. And okay, so this is a very same principle, going to valine and isoleucine. When you go to leucine, there is something very interesting here. And here is what I mean. So this, again, we see another version of the modified part of the TCA cycle. This is a canonical TCA cycle. Okay? So we start with oxaloacetate and you get alpha ketoglutarate. We saw how with +1 you can get to alpha ketoadipate. Okay? It turns out that you can do even fancier things. We saw this, right, in lysine biosynthesis, how from alpha ketoglutarate, you start with alpha ketoglutarate to get to alpha ketoadipate. Well, here, instead of alpha ketoglutarate in lysine biosynthesis, in leucine biosynthesis, we start something even more complicated, 2-ketovaleryate, right? So you mutate the enzyme to make it even bigger and accommodate this more complex substrate. But then, once you do that, then the same mechanism applies. So you bring in acetyl-CoA, you extend by 2, you make the whatever the enzyme is called here. This was homocitrate. I don't even know how. So citrate in the TCA cycle, homocitrate here. I forgot even the name of this intermediate. Then you make the aconitate version, and then you decarboxylate to get what you want in the end, and to get leucine. So this is what. If you need to remember anything from this, is that these are different flavors of the same strategy, right? You modify pre-existing reactions, and you just tiny modifications to achieve the goal you want, but the same chemistry.